First of all, I really do want to thank Hickey for joining us at this hour. It's 11:30 p.m. in Australia, almost midnight. Yes, he has joined us. So we are really grateful to you, Dr. Hickey. With that, I'd like to start our seminar. Bismillahirrahmanirrahim. In the name of Allah, the most gracious, the most merciful, we would like to start Shaping the Future virtual seminar on speed breeding technology entitled Speed Breeding Crops to Feed 10 Billion. Assalamu alaikum, Eid Mubarak. We are on the eve of Eid al-Adha. So if you don't know what Eid is, it is like Christmas for Muslims, a week-long celebration. So good morning, good afternoon, good evening, everyone, depending on wherever you are watching us live on Zoom, Facebook, and YouTube. I'm your host, Sam Shurkar from Bangladesh. Welcome to the seminar. There are three primary ways you can participate in today's event. The first 250 registered people have joined us with the panel via the Zoom app. Others are watching us live on Facebook and YouTube. So if you face any difficulties in one of the platforms, then please consider trying another. For Facebook, please visit tiny.cc/spb04, or you can watch us live from SciencePortabD's official Facebook page at fb.com/SciencePortabD. For YouTube, please visit tiny.cc/SPB03. You can find all the links at SciencePortabD's official Facebook page at fb.com/SciencePortabD. Dear Zoom attendees, please make sure to keep your video and microphone turned off other than the panel. And for panel, please make sure to mute your microphone other than the speaker so we do not get any background noises and participants have no difficulty understanding us. Dear attendees, please put down your headphones for the best ever experience, but still, if you face any difficulties whatsoever, please let us know in the comment section so we can fix them as soon as possible. First of all, I would like to thank our partners and sponsors. Shaping the Future virtual seminar on speed breeding technology is brought to you by Science Porter BD, sponsored by Cell Genetics and Plant Biotechnology Lab. Science Porter BD is a scientific communication platform based in Bangladesh. Members are highly enthusiastic to explore and do science. We communicate and collaborate with other interested groups and peoples who are thriving for a common goal from all around the world. This has been our third international initiative. Our first Shaping the Future virtual seminar was on genome editing entitled The Basics of Genome Editing What, Why Now by Dr. Kutubuddin Ali Mullah, which was a huge success. We had 716 registered participants from more than 20 countries, followed by a successful second one on the production of fourth generation biofuels entitled Microalgal Cell Factories, the Fourth Generation Perspective for Industrial Biofuels by Dr. Pavan Jutur, where we had 453 registered participants from all around the world. Both of the seminars were highly praised by the scientific community. You can watch the recorded video on our YouTube channel at tiny.cc/spb_YouTube. If you want us to host more seminars like this one, then please make sure to like and follow our official Facebook page at fb.com/SciencePortabD. Subscribe to our YouTube channel at tiny.cc/spb_YouTube and hit the notification bell icon to be the first to get the latest updates on our upcoming seminars. I would like to thank all the organizations that have cooperated fully throughout our promotion. I'd also like to thank our print media partner, the World Chronicle, for covering us in the print media. And finally, I would like to thank you, the audience. We were overwhelmed by your responses. We have received 610 registrations from all around the world. Without your continuous support, we couldn't be here today. It would not be possible without the active contribution of Science Porter Bangladesh team, especially the content writers, starting with the team lead, Nazneen Liza, Abi Bakr Siddiq, Abdi Rajak, and Rifat Ahmed. Translators, starting with the team lead, Nazdin Islam, Afriza Tabassim Toma, Israt Khan Enena, Nafiz Yasir, and Nurjahan Akhtar. Social media team, starting with the team lead, Mahua Sharkar, Abrar Hussain Chaudhry, Fairuz Samiha, Fayyad Farhad, Farsan Amin, Israt Khan Enena, Jarif Mehbib Straban, Mahfuz Mao, Majarul Islam Rajib, Maria Meha Prami, Abid Muqtadir Risha, Muhammad Sakawat Alam Faisal, Nazmin Sakib, Nur Jahan Akhtar, Papia Rahman, Rakibul Hassan, Sakib Puyan, Shamima Afros, Sulaiman and Afrida Tabassum Tama for doing an amazing job on social media. And of course, the streamers, starting with Nazrul Islam Akash and Mahfouz Amal for Facebook streaming, Jannatul Firdaus and Mahmoud Hassan Rifat for YouTube streaming. So a big round of applause for you guys as well. Without further ado, let's introduce you to our panel. Let's meet today's event chair, Dr. Abdullah Muhammad Shoal, Professor of Biotechnology and Genetic Engineering at Jahangirnagar University, Bangladesh. Dr. Shoal is a respected authority in the field of biotechnology, but for those of you who don't know him, he is the director of Cell Genetics and Plant Biotechnology Lab that is well known for his high research output. He did his PhD in agricultural biotechnology from Chungbuk National University, South Korea. He had worked as a research collaborator for the International Rice Research Institute in the Philippines. He was awarded the prestigious JASPS postdoctoral fellowship. He had worked as a postdoctoral research associate at the Cell Genetics and Citrus Improvement Lab at the University of Florida. Dr. Shoal has published articles in many reputed international journals. We are honored to have three distinguished scientists from BRRI, that is Bangladesh Rice Research Institute, as our esteemed panelists. Former Director General of BRRI, now working as a national consultant for IRRI, that is IRI, Dr. Jeevan Krishna Bishwash, current Director General of BRRI, and a senior scientist, Dr. Muhammad Shahjahan Kabir, Chief Scientific Officer and Head of Plant Breeding Division at BRRI, Dr. Kandikar Muhammad Iftikharullah will join us in the discussion, along with our event chair, Dr. Abdullah Muhammad Shoal. And now, the attraction for today's event. We are very delighted to have one of the world's leading experts in speed breeding technology, Dr. Lee Hickey, joined with us from Brisbane for this session. Welcome, Dr. Hickey. Dr. Hickey is a world-renowned plant breeder and crop geneticist. Needs no introduction, but for those of you who don't know him, Dr. Hickey is working as the Principal Research Fellow at the Center for Crop Science within the Queensland Alliance for Agriculture and Food Innovation at the University of Queensland, Australia. Dr. Hickey has played a pivotal role in the integration of leading-edge breeding technologies such as the rapid generation advanced technology speed breeding with genomic selection and genome editing. Dr. Hickey's advice to speed breeding crops is sought internationally, and the technology is now adopted by plant breeding programs worldwide. His research outcomes are featured in 60 referred publications, including articles in high-profile journals like Nature Plants, Nature Protocols, and Nature Biotechnology. Dr. Hickey is a prolific science communicator, and his research outcomes have appeared in mainstream media such as BBC World News, National Geographic, and the New York Times. He is passionate about training the next generation of plant breeders and currently mentors 13 PhD students, while 11 of his previous graduates now work for leading plant breeding companies and high-profile research institutes around the globe. Enough intro, let's dig in. Let me give you a rough overview of today's event. First, our speaker, Dr. Lee Hickey, will talk about speed breeding technology in great detail. Then today's event chair, Dr. Abdullah Muhammad Shoal, will greet you with his message. Then our honorable guests will join us in the discussion. In the meantime, our team will collect your questions and we will go for a Q&A session shortly. So make sure to leave your questions in the comment sections of Zoom, Facebook, and YouTube Live ahead of time. Dr. Hickey, the floor is all yours.
Great, thanks a lot. Yeah, thank you very much for this great opportunity to speak to you all today and share a bit about speed breeding technology and how it can be integrated with other breeding technologies to accelerate both crop research and crop breeding. Because we all know, as plant scientists and breeders, we all face a great challenge to feed our future populations. To kick things off, I think I'd like to start by playing a short video as an introduction.
Population is still growing. By 2050, we'll have something like 10 billion people on this planet. So it means we've got to produce more food. But we've got to produce more food under climate change. In Australia, we're facing one of the worst droughts we've ever seen. Farmers in Europe, on the other side of the planet, are also dealing with drought and heat. So we really need to speed up the development of more productive crops in the face of climate change. Plant scientists and breeders all around the world working on discovering new traits and genes for drought adaptation or heat tolerance that can be deployed in future varieties. The problem is, it just takes so long to put those traits into varieties for farmers to grow. So speed breeding can really help fast track this process and deliver more robust crops to farmers sooner rather than later. Normally, this technology can be quite expensive to set up large-scale facilities. So we wanted to empower the plant scientists with a small budget. A latest research paper between the University of Queensland and the John Innes Centre in the UK, we've reported the protocols on how to build your own speed breeding cabinet or DIY speed breeding.
We've also built a miniature desktop cabinet with bits and pieces that we got off the internet. It was very cheap. It only cost £700 or £800. And this tiny little cabinet now allows us to trial a new crop or a new cultivar of a crop and look at a new trait. And once we've optimized the parameters, we can then scale it up in the glasshouse.
We're hearing more and more from institutes around the world establishing speed breeding facilities to help speed up their plant research or breeding. And we're pretty excited that maybe one day speed breeding will really deliver some of our future varieties for our farmers.
Okay, so what we're looking at right now is one of our speed breeding rooms at the University of Queensland. Basically, this is a temperature controlled facility fitted out with the latest LED lighting system, and where we can actually control the environmental conditions to achieve rapid generation in advance. To accelerate all of our crop research and breeding interactions with various companies in Australia. The inspiration for this speed breeding technology actually came from NASA-funded research that was actually done way back in the 80s, or at least started back in the 80s. And they were aiming to grow wheat in outer space. The way that they were doing it was, well, they were exposing the plants to continuous light and controlled temperatures to accelerate plant development. So this tricks the plants into the reproductive phase much faster, and you can produce a really quick crop of wheat. And so you can see this video playing on loop here is actually the video of the first wheat crop grown in space at the International Space Station back in 2018. And so you can see this approach or idea is really not new. It's been around for some time, and my colleagues at UQ started applying this and developing this back in 2003. However, I would say that interest in speeding up our crop breeding to develop better crops has really peaked, I would say. Over the last few years, I've received hundreds and hundreds of requests from scientists and plant breeders around the world really wanting to know how exactly we implement speed breeding. And so, there wasn't really a lot of information out there about how to go about setting up a facility and implementing this. So this really inspired us to publish and make available these protocols in a lot of detail to hopefully enable or inspire widespread adoption of this enabling plant breeding tool. And so we teamed up with researchers from the John Innes Centre and the University of Sydney, and we published our protocols for the first time in 2018. And we were very lucky. We actually, the article was featured on the cover of Nature Plants, which is not very common for a plant breeding tool, and which was really exciting. And before I go any further, I have to acknowledge some of the key people involved in pulling all these protocols together, and that's Dr. Brando Wolf at the John Innes Centre, and of course the two co-authors of our two protocol papers, now Dr. Amy Watson and Shreya Ghosh from the University of Sydney. Amy was my PhD student. Now she's in the UK. In a nutshell, this is what we can do with speed breeding. We can grow 6 generations of wheat per year instead of just 2 or 3 in a regular glasshouse. And I say 2 or 3 in a regular glasshouse because it really is variable. It depends on the season, and the environmental control is not very good. In speed breeding, we're using 22 hours light and only 2 hours of dark each day. And during those 22 hours of light, the plants are growing at 22 degrees Celsius. And during that 2-hour dark phase, where the plants are resting, the temperature drops to 17 degrees. And we find that this rest phase improves the quality of the plants without compromising speed. Just to show how fast these plants actually grow, because seeing is believing, right? We captured this time lapse in collaboration with our partners at the Earlham Institute. And so there you have speed breeding spring wheat on the left, and wheat in the same wheat variety going under regular glasshouse conditions on the right. And the speed breeding plants have already flowered. They're now producing grain, starting to mature, and it's harvest time. And unfortunately, we don't have time to finish the video because normal breeding is just too slow. So this same protocol that was originally developed for wheat actually works really well for a whole bunch of different long day and day neutral species. So crops like bread wheat, durum wheat, barley, chickpea, oat, canola, pea, quinoa, grass pea, even peanuts, lentils, and potatoes. And so it also has benefits for model species like Brachypodium and Metacargo in terms of reducing the generation time. And these pictures you're looking at here really provide a visual illustration of how fast the plants actually develop in speed breeding, because these plants were stolen on the same day, but the plants on the left going under speed breeding and the plants on the right going under a regular glasshouse. And you can see after 3 to 4 weeks of growth, at flowering time, these plants were photographed together, and you can see how amazingly different they are in terms of developmental stages. As a follow-up publication, we actually provided a step-by-step guide for setting up small and large-scale facilities, and this was published in Nature Protocols. It was a huge effort to pull this together. There's literally so many tables and figures of information. It provides troubleshooting, things that we often encounter, problems, tips on how to go about resolving them, and even more crops. So it's a really great resource if you're thinking of setting up a speed breeding facility. The bird's eye view you're looking at there of the video is actually one of the large-scale speed breeding facilities at the John Innes Centre. And the John Innes Centre actually had to go and create additional space because of all the researchers there wanted to use speed breeding to speed up their research. In this protocols paper, we also outline how to set up your own benchtop speed breeding cabinet. And so this is really suitable for labs or researchers with a tight budget. And I guess really suitable for people who are doing transgenic research or perhaps integration or backcrossing work, and you're really only dealing with a very small numbers of plants. But plants where you require adult plants for crossing or perhaps phenotyping adult traits. And so this is really thanks to this bunch of early career researchers you can see here from the John Innes Centre. They came up with this amazing idea where literally you can order these bits and pieces from the internet, mostly Amazon.com, and construct your own cabinet from scratch. And so all the code is available through this publication. This thing is even programmable through a Raspberry Pi. You can control temperature, the photo period and lighting regimes, etc. So really, really amazing. We actually ran a workshop, a speed breeding workshop in Kenya last year, and so you can see this video and the photo there just showing the end product of the workshop, the participants actually got their chance to build their own cabinet from scratch. And I think we built 3 cabinets in total, and 3 lucky participants actually took home those cabinets. So, but while the cabinets are useful, what I think is more useful in building these cabinets for this workshop was actually learning and absorbing and thinking about what are the key elements that we need to control in order to apply speed breeding. Things like temperature, photoperiod, light quality, nutrient supply, water supply, etc. When it comes to scaling up for crop breeding programs or big research labs, this is often a good option where you have existing infrastructure like glass houses. This is essentially what we've done at the University of Queensland. We already had reasonably good glass houses, so we were able to just fit them out with LED lighting systems for speed breeding. And so all the details about how our facility is set up is all provided in this guide. Also, the setup for the John Innes Centre is also there. So I must stress that there's no one size fits all when it comes to setting up speed breeding facilities. This whole approach is very flexible and it can come in very many forms and shapes. So you can really think of this as a guide and really adapt this to your own environment and system. When it comes to scaling up, I really like to, and particularly for breeding applications like single seed descent, I really like to think of this as creating plant factories. You want to produce a plant for as little cost and as little resources as possible, just a few cents ideally. And so we can grow plants at very high density to reduce this cost per plant. And you can see here an example for barley grown at different densities. And even at 1,000 plants per square meter, these spikes are producing up to 10 seeds each. So that's 10 seeds per plant, which is more than enough for single seed descent applications in breeding programs. But we've actually grown these plants at even 3 times this rate of 3,000 plants per square meter, and we can still achieve seed from most of those plants. So when we weigh up different strategies for developing inbred lines from across, there's pros and cons of these, and I'd like to run through some of the options we have, and let's look at the time scale of adopting these approaches. So here's the traditional approach for inbreeding, where we're doing 1 generation a year from crossing to development of inbred lines, and let's call them F6 lines. And you can see if you do that, it takes 7 years. It's pretty simple. I must say that a lot of companies around the world are going much faster than that these days, and it's common, or not common, but it still does occur for some crops that have biological constraints or breeding programs that have very limited resources. This is still going on. Shuttle breeding is a technique that's been widely adopted now, first initiated in Simmet by the famous Dr. Norman Borlaug, of course, and it's usually successful for maize as well, shuttling materials from location to location to get 2 generations a year. And it's simple math. You can do it in half the time. Speed breeding, well, you can do it even faster, right? And depending on the species, you can grow between 4 and 6 generations a year. So, you can do it quickly. And I would say at a similar rate to double haploids, which still requires growing a few generations to produce those. And while double haploids have had a huge impact in the plant breeding world, particularly for winter wheat and maize, there are technologies like any technology, and I'd like to point out a few of those. Certainly there's very limited opportunity to perform early generation selection, like in the F2, etc., F3, as they go through the inbreeding process. You get generally lower rates of recombination, only 1 recombination rate compared to every generation you the single seed descent process, and of course the efficient across crops or germplasm. Some materials are compatible with double haploid systems, and of course it's laborious and can be quite expensive. And I know it can be up to about €12 per double haploid line for wheat in Europe, and about half of that, €6 for barley line. And so when you this equation before, essentially this is how a plant breeder achieves genetic gain over time in the breeding program. In the upper components, the plant breeder is really trying to increase selection intensity, selection accuracy, and genetic variance to some extent. And of course, at the bottom, the denominator of the equation, the really powerful part to tweak is time and essentially years per cycle. And that's where shuttle breeding comes in. And of course, speed breeding acting in a similar way there to reduce the length of the breeding cycle. So what about short day species? I've spoken a lot about day neutral and long day crops. What about short day crops? Can we speed breed those as well? Well, the short answer is yes, we can. We can literally speed breed any species. It's just a matter of optimizing the environmental conditions to achieve rapid generation advance. And what really awoken me to this reality was I heard about some breeding efforts in New York. It was American chestnut tree, which is a huge tree. The researchers there actually figured out that you could essentially speed breed American chestnut by exposing the seedlings to high intensity light. And this initiated flowering within 12 months. And so you can actually perform a cross within 12 months, which is really amazing. And so if you can do this for American chestnut, I reckon you can do it for just about any species. And so I guess this figure really summarizes some of the key aspects that you can think about optimizing in terms of the conditions for speed breeding protocols. And the two most important for me, really, it comes down to day length and temperature. And once you've optimized those two things, here's some of the other aspects you can start to tweak. Of course, light quality is really important. Nutrients is important. The delivery of certain nutrients at different times, even withholding nutrients can have a substantial effect on flowering time and maturity in some species. For example, nitrogen, starving, or not necessarily starving, but withholding nitrogen supply can also accelerate plant development for crops like wheat. And so also things like plant density is really important for competition, and it can have different effects in different species. High density for crops like wheat and barley can accelerate time to flowering, but high density for crops like sorghum can actually have an opposite effect. So it's rather interesting, but these things are quite species specific when you start to really optimize the conditions. And so I've been working on some of these short day crops together in a collaboration with ICRISAT. This is part of a Bill and Melinda Gates Foundation, and ICRISAT have this mandate. They're actually one of the CGIAR breeding programs, breeding crops for Africa and Asia. And this includes sorghum, millets, groundnut, chickpea, and pigeon pea. So crops that actually I had very little experience in before starting to work with ICRISAT about a year or so ago. And so I'm learning a lot as well as part of this collaboration. But this project, you know, we've essentially been developing speed breeding protocols and facilities for these crops. So it's been a a great and rewarding experience. So far, through several strategic experiments, testing different combinations of temperature and photoperiod. Essentially, this is the best protocol that we have at the moment for sorghum and millet. You can see it's much warmer temperatures for starters. These are tropical crops, so 35 degrees during the daytime hours and 25 degrees during the night. And it's not long photoperiod like wheat. It's a short photoperiod of 10 hours, which works to trigger early flowering in these species. And so using this protocol, we can achieve 4 generations of sorghum and 5 generations of millet per year. This is a work in progress, so we're continuing to tweak other aspects of the protocol, and hopefully we can achieve somewhere in the order of 5 to 6 for these species once we've optimized the process. But certainly right now, this protocol is quite efficient in achieving 4 to 5 and very valuable in terms of comparing this to field-based generation advance. When it comes to scaling up these systems, just like wheat and barley, it can be done for millet and sorghum. Here's a tray of millet, a segregating population, grown at 300 plants per square meter, and on the right, sorghum, grown at 600 plants per square meter. And I'd say this is a little bit too crazy for sorghum. It's a lot larger plant, and like I said, at this high density, sorghum seems to the time to flowering seems to be longer at this high density plantings, and so 300 plants seems to work quite well for millet and sorghum in general. We've been making some good progress in pigeon pea, and working with colleagues at ICRISAT, we've shown that you can actually adopt this early pod harvest technique to achieve 4 generations per year. So it's a similar strategy to the technique that we developed in wheat and barley, where we actually harvest the seeds prematurely, and using germination protocols, we can get good germination from that premature seed. And so we're now optimizing these protocols for both photoperiod sensitive and insensitive types, which is really important for their breeding operations. Recently, there's been new protocols published for short day crops, soybean, rice, and amaranth. And I was really excited to see this paper come out. It's essentially they're using very similar, or they've reported a very similar protocol to the one we've developed for sorghum and millet, and that's using a 10-hour photoperiod as well. And you can see here some of the pictures from their study, where they found that they actually did a whole series of these beautiful experiments testing different compositions of wavelengths on plant growth. And so they were looking at 2 main factors. So one, the size of the plant. So could they restrict the plant size and keep them short and sturdy, which is good for packing more plants in. And of course, time to flowering. And they found that if you actually withhold far red light for soybean, you end up with short, sturdy plants that flower fast. And in rice, it was actually the opposite. They actually needed a light enriched with far red spectrum to achieve rapid flowering. So for me, this was really interesting that you can actually tweak and optimize photoperiod compositions, wavelength compositions for each of these species and manipulate plant growth. So a lot of room for optimization there, and I think for a lot of the other species. And so one of the reasons why they started developing these speed breeding protocols, this was work done at the University of Hohenheim in Germany, was because this was their traditional approach. They had winter nurseries and the main season, and so often every 2 or 3 years, the winter nursery would fail, and so it wasn't very reliable for their breeding operations. And so now with speed breeding, they can achieve 5 generations a year, quite routinely and robustly. So just wanted to talk a little bit about speed breeding facilities and the different shapes and forms that they come in for the different crops. So some commercial examples. So here's an overview of a facility of BASF, the spring wheat breeding program for North America. And I have to thank the breeders and the companies for sharing this information. I think it's a great initiative of them to make this information available and really inspire new ideas of how we can adopt these tools. Here you can see this is essentially a speed breeding warehouse. This is wheat growing on multiple levels. So 3 different levels up because they actually figured out that it was easier to go upwards to control temperature in their very extreme environment that has very harsh winters. And so in doing it in a shed was more cost effective than having an actual glass house. Because in a glass house, you have sun coming through with heat load during summer. And of course, it's much harder to heat the environment in harsh winter times. Another example is this is a facility, work in progress in Ethiopia. And this is where they have existing glasshouse infrastructure. And we're fitting these rooms out with heliospectra LEDs and upgrading the temperature control, the air conditioning system. And so you can see here Zerihun, one of my PhD students there in Kalumsa, measuring out the room. And so we've got all the equipment ordered, and unfortunately COVID has delayed the installation process, but hopefully we'll be able to get there and set all this up very soon. Another example, this time for speed breeding pearl millet, and this is in India at Corteva, and I was lucky enough to actually visit this facility outside of Hyderabad, and it's just a beautiful facility where they've actually moved all of their breeding operations from outdoors to indoors into this speed breeding facility. And the reason they did that is because of cost and efficiencies. They went from managing a very large team, turning over materials in the field, to a very small refined team of 5 or 6 people that manage the whole operations in this speed breeding facility. And they're producing somewhere in the order of 50,000 lines per year in this facility. So, 4 generations a year, high density plantings, a lot of automation and streamlining to really reduce the costs and improve the efficiency. This was really critical for their breeding program. And of course, it's Corteva, so they have fabulous marker-assisted selection programs, and this is all fully integrated in the speed breeding process, where all single plants are tracked along the way during line development using barcoding and that sort of thing. So as part of the ICRISAT Bill and Melinda Gates Foundation project called Avisa, Corteva are actually partners on this as well. And so they also had a great deal of input and actually helped design some of the facilities that we've recently constructed at ICRISAT. And so here you can see one of them under construction. And this is essentially a similar facility to the Corteva one, where they did the millet speed breeding. And essentially how this works is you have blackout curtains installed that actually block out the light. And so these can be set on an automated timer. So you can actually impose 10-hour photo periods all year round, very robustly. And so with some limited temperature control, evaporative coolers and fans up the other end, you can make sure the temperature does not reach above 35 degrees. And so this facility is actually finished and completed early this year. So to take a look through some of the facilities now, here on the left, you can see some of the existing glass houses that were actually fitted out for speed breeding chickpea and peanuts. In this room, they can actually grow 3,000 chickpea inbred lines in a single year. And so you can see the automated irrigation system there. The temperature control was upgraded, and of course the LED lighting system. On the right, we're now walking through the facility we saw on the previous slide under construction, and this is the end product. So this is a facility suitable for speed breeding short day crops, automated irrigation. The blackout curtains are drawn. You can see it's completely black in the middle of the day, and it works beautifully. And in this room, they can actually grow up to 50,000 inbreds in a year. So this is a lot more inbreds than you could possibly imagine field testing. The cost of field testing 50,000 lines is, of course, very expensive. Another example, we go to rice and the nice RGA system at Erie. I think this is a great example which really highlights that cost and efficiency is really important. So at Erie in the Philippines, they have this beautiful RGA set up for the glasshouse, and you can go a little bit faster than the field, but in the field, it's a lot cheaper. So you compromise speed a little bit, but they can achieve 3 generations a year for a lot lower cost. And so this means they can grow a lot more lines and play the numbers game from a plant breeding perspective. And even in the field, from what I understand, they apply a lot of tricks in terms of withholding water to accelerate the generation advance. And so this is part of the process. And so I really want to thank Josh Cobb for providing this slide and information and showcasing their really nice work there at Erie. Another example from Akada, I was really impressed by this. This was provided by Aladdin, and this was set up by him over there in Lebanon. And essentially they've set up speed breeding in the field, which is really, I couldn't believe it when he told me this. They've bought LEDs from a local market, which costs about $30 each. So really low cost from a local supplier. And you can see they're just hanging from this structure over the field, and can really achieve speed breeding at a super low cost all year round. And I believe they're growing 4 generations a year and avoiding the really cold months there in Lebanon. So like I said, speed breeding can come in many shapes and forms. And so here you can see, perhaps this is actually an artist's impression of what maybe a speed breeding capsule could look like. We came up with this for a review we wrote for TAG. I really just throwing around ideas about how we could deliver this technology to breeding programs with limited resources, perhaps in developing countries, and really take speed breeding off the grid and deploy it remotely. And this is essentially a shipping container fitted out with the solar panels to support the energy requirements from the lighting systems and cooling systems. And essentially, you wouldn't have to grow 6 generations a year, even if you could grow 1 or 2 extra generations in the off season. This sort of tool would be very impactful in a breeding program over a long period of time. And while this seems like a dream, it's really happening. So this is Stuart Kemp in Victoria, and I worked with him to set up this speed breeding facility he has down there. He literally built this from scratch out of a shipping container, decked it out with LED lights. And just as an example, if you're thinking about a crop, well, for chickpea, you can grow 15,000 inbred lines per year, just in this very limited space in a shipping container. But of course, Stuart doesn't breed chickpea. He breeds a whole range of forage species down in Victoria, and he's actually working on 12 different forage species. You can see here under the lights. So it's helping to accelerate his program. Just wanted to talk about some examples on how this tool can help pre-breeding activities, which a lot of us in the public sector are involved in. As we all know, our crops have gone through a huge genetic bottleneck through domestication and, of course, now modern breeding, since it started 100 or 200 years ago, depending on the species. And so, you know, we can go back to land races or wild relatives for new allelic diversity, but this brings its own challenges in that we have to separate the useful genes from the junk and deleterious traits. And so this requires many cycles of backcrossing to introduce the desirable traits into modern elite cultivars. And so this can be a rather time-consuming process. And so here's just an example to illustrate this. Here's an example for barley, where we're targeting 4 different traits from land races. So these are 4 different disease traits. So leaf rust, scald, barley yellow dwarf virus, and netform net blotch. 4 genes on 4 different chromosomes from 4 different donors. And so we can take this integration approach through backcrossing, a series of backcrosses, some gene stacking steps, and essentially we can create ultimate stack, super barley with all enhanced traits. But you know, this takes 8 generations, and if we were to do this in the field, you can see it's going to take 8 years to achieve this goal. In a regular glasshouse, you know, you would never do this in the field, by the way. In a regular glasshouse, it would take half the time, 4 years. In speed breeding, it actually makes this whole process worthwhile and achievable now very quickly in just 1.5 years. So it really is opening up a whole new gene pool for breeding. We can introduce traits quickly and move these traits around. And so we're doing exactly this. In my team, one of the germplasm collections that we're working on mining useful genetic diversity from is this Vavilov wheat collection. These are, you know, land races and historic wheats collected by Vavilov and his colleagues from around the world prior to modern plant breeding. And so, you know, these are some of the team here. You can see Eric, Delaney, Ravinja, and so they're working on a lot of disease traits. And so while they're using speed breeding to set up new mapping populations and actually identify the genomic regions where these traits map to and develop useful DNA markers, they're also backcrossing these traits in parallel. So even within a time frame of just 3 years in a PhD program, they can effectively dissect the genetics of a new trait, develop diagnostic DNA markers, and develop elite materials with that new trait. And so this was never really before possible. Another example I'd like to talk about is genome editing pipelines. And genome editing is really shaping up as a powerful tool for plant breeding. And the traits that we could target are really unlimited. We're talking about quality traits, plant architectural traits to improve adaptation, disease resistance, and even, and this is a really neat study, rapid domestication, where this was a study in tomato where they edited multiple domestication genes simultaneously to basically create a new domesticated tomato, which is really amazing. But while this is really promising, there are still many bottlenecks associated with this technology, particularly from a plant breeding perspective. And so I'd like to talk about some of those. So firstly, we need cloned genes or gene targets for this technology. Secondly, it's rather lengthy passages through tissue culture, and they require specialized labs to undertake the genetic manipulation with Cas9 and the guide RNA sequences. And let's face it, anything that goes through the lab is a bottleneck from a plant breeding perspective. And of course, this whole process is often limited to a very small number of non-elite genotypes. A plant breeder really wants to edit any variety or any line that he or she wants. So in our speed breeding Nature Plants paper, we highlighted how this tool could actually be integrated to accelerate transgenic pipelines. And so here you have standard transformation timeline for barley, where you actually grow the barley plants from seed to produce immature embryos. You then harvest those embryos and transform them to produce explants, and then you grow on those plants and obtain T1 seed. And so, this is a rather lengthy process. So what we did is we grew barley plants under speed breeding conditions to rapidly obtain embryos under speed breeding. We transformed those embryos and grew on a batch of explants under speed breeding and regular glasshouse conditions. And you can see the rate of developmental differences are quite contrasting here. For speed breeding, much faster. And what does this do on the timeline? Well, it saves about 3 months because it shrinks both ends. You can actually do this faster. And so we've got to stop thinking about these tools as independent things. We've got to start to integrate these tools and technologies, and that's where the real efficiencies lie. And so what does this mean? Well, we can validate new traits faster. CRISPR and transgenics is a fabulous tool for trait validation and understanding gene function. We can deliver these new traits into farmers' fields faster, where the technology is accepted. And of course, we can publish that Nature paper faster. Very important for some people. So we got thinking with some colleagues of mine, what if we could take CRISPR out of the lab? That would solve a pretty big problem from a plant breeding perspective. And what if we could remove the genotypic limitations of this tool? And what if we could edit any line we wanted? And so we came up with some ideas, and this is really a work in progress. We have several PhD students working on this at the University of Queensland, and I really have to thank Professor Ian Godwin for a lot of these ideas and concepts. Here's a summary of our concepts, basically, that we're working on. And this was published in our review in Nature Biotechnology last year. If you're interested in finding out more, you can go and have a read. But very briefly, this is sort of what we have in mind and we're working on. This is all happening in a speed breeding environment. So we're doing this out of the lab, directly in the speed breeding system. And there's different ways we can go about this potentially. So here we have CRISPR-ready Cas9 seedlings, which may already carry the Cas9 gene. We can deliver guide RNA into the speed breeding system, perform edits, identify the plants that actually have that edited gene or trait through trait screening. And in this case, this is just an example for a disease resistance gene that has been edited in sunflower. But you could think about these traits being any trait in any crop. And once the edited plant has been identified, with Cas9, we can continue to do cycles of editing for other traits. However, if we wanted to segregate out the Cas9, we can. And if we do that and retain the trait, this plant is actually considered non-GM in Australia under the new regulations. And this plant with the new trait can enter field trials just like any other plant. So this is really exciting. The technology to deliver the guide RNA in this manner is a little more complicated than a spray bottle worth $1 or $2. There's lots of options we're exploring. It's very important that the guide RNA and the edit occurs in the plant, so it is inherited, the next generation. It's critical. And so some of these techniques we're exploring. There's already methods out there for tissue culture-free delivery, and so we're exploring some of these at the moment. So what we're trying to do really is transform this genome editing from a biotechnology into a highly flexible and efficient breeding technology. And we really need the whole community to be thinking in this way so we can achieve this goal together. Now I'd like to talk a bit about genomic selection and how speed breeding can also be beneficial in these predictive breeding approaches. So just another way to visualize a breeding cycle. This one is cyclic in nature here, as breeding cycles are. And so here you have the crossing, you're doing the inbreeding, then those inbreds are evaluated in the field, which once you identify the winner among the pack after extensive testing across environments and years, you can release a variety. And of course, through all that testing, you generate data to select the best parents for the next cycle of crossing. And so genomic selection kind of works like this. You don't need to actually test the inbreds to make decisions about which are the best inbreds to use for the next breeding cycle, or perhaps better set of inbreds to enter the field testing program. And of course, you don't get out of testing, you still have to do the testing. And in fact, you might be testing a better set. And of course, this is needed for variety development as well. But of course, this data that you generate is also important for retraining algorithms. And so what happens when you introduce speed breeding into this equation is that you can actually do this inbreeding much faster. And so you can crank these cycles even faster. So this is really promising. To explore the potential of adopting this SpeedGS approach, combining speed breeding and genomic selection, we teamed up with researchers from CIMMYT and in Germany, and using real wheat data sets from the CIMMYT breeding program and breeding schemes, we actually did a series of simulations. And so here you can see, after 30 years of breeding, the conventional approach and SpeedGS, much better in terms of genetic gain and varieties that have a higher yielding capacity. And so this equates to 34% extra genetic gain. And my colleague Kai, for a bit of fun, he did the sums, and he worked out that if you use this breeding scheme, you would be generating varieties that yield higher on average by more than ½ a ton per hectare after 30 years. And this would equate to more than 1,000 loaves of bread extra per hectare. And so, you know, we talk about these technologies helping to develop crops that are going to feed the world. Well, I think this is a great example where if we bring these technologies together, fuse them together, we can achieve these sorts of goals for the future. And so I guess we're not just simulating these things, and talking about it, we're actually doing it. Here's Amy Watson, she's standing in the field of the first wheat lines developed using the SpeedGS approach. However, as a university and researcher, we can really only do this at a small scale. It's very expensive to do multi-environment trials and really validate these breeding processes. So it really makes sense for us to team up with industry to try to validate these breeding approaches. And so we're doing that through a series of projects. This is an example, working with a company called Longreach in Australia, one of the breeding companies. And we're really taking genomic selection to the next level. We're not only combining it with speed breeding, but we're also applying it in a totally novel way, using artificial intelligence algorithms that actually not only determine which are the individuals with the highest genetic merit, but the individuals that are the most optimal for crossing, which really improves efficiencies. And so this is crazy ideas actually come up by Professor Ben Hayes and Kai. Ben was actually the co-inventor of genomic selection, originally applied for dairy cattle breeding. And so he's got some really crazy ideas. So I'm really learning a lot from these guys as well. And so in summary, so just to highlight the value of these tools to speed up breeding cycles, here's the traditional breeding approach where you have the crossing and inbreeding phase, the testing for yield, disease quality over many years and locations, and of course, seed increase and release of those new varieties. And so using speed breeding, we can really cut down this whole crossing and inbreeding phase. And there's big potential to increase the rate of genetic gain, depending on how fast you're already going. And of course, for the breeding cycle, you can start this sooner because you generate data on those selection candidates earlier, and you can make decisions about the next breeding cycle sooner. When you combine speed breeding and genomic selection, well, you can actually do the predictions on those inbreds, so you don't have to wait to predict them to start the next breeding cycle. And so again, this has a double whammy effect on genetic gain in a breeding program. Some take-home messages. Well, I hope you can see that speed breeding systems can be developed for any crop, and there are applications to accelerate all aspects of research and breeding. So discovery research, pre-breeding and breeding activities. There's benefits of growing plants fast. We're, all of the time as plant researchers or breeders, we're just waiting for plants to grow. So I think this is a really critical thing to pair with the latest genomic technologies. Speed breeding platforms are really becoming very common in the private sector, and we really need equivalent facilities, efficient facilities in the public sector to exploit this tool. And to do that, reducing the costs associated with implementing this technology is critical, and that's where streamlining operations and setting up facilities in the right way is really critical. But we've got to think about this as a return on investment as well. Time is money, and when it comes to developing our crops for the future, this is really critical. And integrating the technologies can really improve breeding pipelines. I didn't really talk too much about it, but you can imagine it's very easy to combine marker-assisted selection and speed breeding during line development. So that's an obvious one. But of course, I talked a bit about combining genomic selection and speed breeding. Huge gains to be made, but there are challenges with implementing these technologies, particularly genomic selection. It requires databases, data management, and major operational changes to a breeding program. But these things can be achieved. It just requires a lot of steps for a breeding program to get to that point. I really want to thank a huge army of people from around the world who really contributed to all of our speed breeding protocols and a lot of the research I talked about today. Thanks a lot to our funders as well, the ARC, the Australian government, and also the GRDC. This is essentially money from our farmers that gets reinvested back into grains research. Thanks very much.
Thank you, Dr. Hickey. That was a very captivating presentation. And now I'd like to request our event chair, Dr. Abdullah Mohammad Shual, sir, to share a few words with us about this new platform that he has started. Dr. Shual, sir.
Bismillahirrahmanirrahim. It is indeed a great pleasure and honor to be here in this event, International Seminar on Speed Breeding Crops to Feed 10 Billion, jointly organized by Cell Genetics and Plant Biotechnology Lab and Science Portal Bangladesh. We are really lucky to get our speaker, renowned scientist in speed breeding, Dr. Lee Hickey, and of course we learned a lot from him. Definitely we have lots of questions to him. Please bear with us to answer almost few questions at least, I think, because I got tons of questions and then maybe we can discuss some of those. Honorable guests, we have Dr. Shahjahan Kabir, Director General, Bangladesh Rice Research Institute, Dr. Jibon Krishna Bishwas, ex-Director General, Bangladesh Rice Research Institute, Dr. Khondokar Iftekharuddollah, Head of Plant Breeding, Bangladesh Rice Research Institute, representative from Research Institute and other institutes, and distinguished participants from home and abroad. I welcome all of you who are directly connected with us in Zoom and Facebook, YouTube Live. My sincere thanks to Mr. Saim Sarkar, host of this program, and his communication team from Science Portal Bangladesh for the arrangement of this whole program. Science Portal Bangladesh members, Cell Genetics and Plant Biotechnology Lab members, Department of Biotechnology and Genetic Engineering, Jahangirnagar University Authority, BDDN Network to support our program. Assalamu alaikum, good evening, good night, good morning to all of you. I will not talk much, but directly I will go for panelist with question and answer, and also we will have the discussion parallelly. I am requesting to open the mic and video Dr. Jibon Krishna Bishwash sir, Dr. Shahjahan Kabir, Dr. Iftekharuddollah, and I am requesting Professor Tofazul Islam sir. I saw he is here, so if you don't mind, so please join with us in the discussion panel. Dr. Iftekhar, are you there?
So, thank you very much. We have been highly enlightened, enjoying the seminar delivered by Dr. Lee Hickey, and we are happy to join in the panel discussion.
Okay, thank you. Thank you, Editor Iftekhar. I would like to take a moment to thank all of you and all of our participants. This is Eid season, so Eid is like Christmas for Muslims. So we had a lot of problems getting our streamers ready. Without your continuous support, we couldn't be here today. And now, I would like to request our honorable guest, former Director General of BRRI, Dr. Jibon Krishna Bishwash, to share a few words with us, and then we will go for a Q&A session shortly. Dr. Krishna Bishwash.
Yeah, thank you very much, Nasratullah Sarkar. So today,
Thank you, sir.
I am very much excited that I was chosen as a panelist, but I am very much shaky because, in fact, I am not a breeder, plant breeder by profession. I am neither a breeder nor a molecular biologist. Simply, I was a rice worker at BRRI more than 33 years, and in fact, I worked as a plant physiologist over the early stage physiologist. So, I was expecting our DG, isn't DG? Shahjahan is here?
Yeah, yes.
Okay, thank you, Shahjahan, because I feel happy if he is here. He is now the real person who is just driving seat in our BRRI. So I am happy that our head of plant breeding, Iftekhar, is here, and Partho Sharuthi, and as well as one of our young scientists who is, I saw him, young scientist, Mr. Abu Said, all of them are breeder. And I think Partho and Iftekhar, they are now working. They are related with the transforming rice breeding system at BRRI, and I think they could share their experience because they are the one, in fact, working on rice in the RGA methodology and others technology. And they are the one maybe quite more or less successful to reduce the time of developing rice variety from 14 years to around 7 to 8 years. So I like to thank Professor Lee Hickey. As I was not a breeder, so I didn't hear about you. Sorry for that. But after getting your name, I try to know about you, and I have got some of your papers in brief and in short. And I found, and today, just after getting acquainted with your works, I found really it is exciting, especially in case of wheat. You have 6 breeding cycles a year, so you could develop maybe a lot of wheat varieties. But in our case, rice, because we are dealing with rice, maybe our breeders, they will explain that in our case, maybe following the RGA methods, our recent breeding strategy, maybe they have reduced the time, maybe 3 cycles a year, because we have winter, we have summer like this, and we have our facility was not so improved before, but our Partho and Iftekhar, they are working as well as in the field RGA method, as well as in the control conditions they are working. Maybe we have also the facilities of particular study methodology. In BRRI, we have started just shortening the screening time to develop a particular sensitive variety long ago, maybe in the 1975s, mid 1975s. Some of our big famous scientists, they tried to develop a HYV, range-fed lowland rice varieties. Because in our country, especially in Bangladesh and the eastern part of India, in the West Bengal as well as in Assam, maybe, we have a rice variety called Amon. Amon is safety and is in fact it is photoperiod sensitive variety. When you will be transplanting, we could harvest in the month of November, and it will be month of December, and it will be flowering in by the first week of November. That is, it is short day plant, and they need day length around 10 hours. So, but in ED strategy, they have developed the photoinsensitive variety. But in our case, we need a particular sensitive variety. So in our case, we need a system how to screen particular sensitive variety. Because if we want to screen particular sensitive variety, maybe we have to wait just only one time we could screen out, because we have to wait for 10-day period. So that's why our scientists developed a technology that is covering by black cloths. So it was quite helpful, and we could do that several times even in a year. So that was, we have done those things, and even we have developed another technology. That is, we used to call it, because without covering a rice crop, we could do that. If we could transplant the transplanted amul rice, which is originally scheduled to be transplanted in the month of June or July, but we could transplant that variety. And if we transplant in December, maybe it will flower partially. Partially from that, because we have the data, maybe 1%, 2%, if it flowers partially, then we could we have a conclusion that maybe it could be 100% particular sensitive varieties. That's the way we have tried to do maybe screen out maybe more than one times a year to develop particular sensitive varieties. Then nowadays our colleagues Partho and Iftekhar, they are screening out in the control condition as well as in the field condition, and they are quite successful. Maybe they have maybe 3 to 3.5, no, 2.5 maybe. 2.5 times they have already already the breeding cycle they have reduced. So I think they have developed some of the varieties and then considerations. Maybe they will explain those things. And with this, maybe I am not expert. I was just a research manager in my institute. So or a rice worker, severe rice workers. So here I am very happy. There are scientists from different parts of the country, different universities, as well as even from the Africa. One of our colleagues I could see, maybe some from India, and of course some from other countries. They are. I am with them. I am happy. You see, there is one of the famous professor and scientist, Tafajjul Islam. Of course, Abdullah Shuaib is very, very famous. I know him. We are related to doing something different from, not different, maybe just to working with what is we call TIFULA, that is advocacy for agrobiotechnology technology, something like that. So I have a good relationship. Though I have retired, but I have some affiliation with other organizations. I was with IRI, but now no more with IRI. But I am teaching in university. Just simply I am teaching the undergraduate students. So I am still doing something. I am happy that, and I have some communication with others also. Sometimes I am writing articles in the popular articles in the newspapers. And that's the way I am maintaining the communication with my colleagues. And with this, I want to conclude my speech, and I also like to thank Lee Hickey and Abdullah Sohail and others. Thank you very much.
Thank you, Dr. Jivan Krishna Bishwash, for highlighting some of the key aspects of plant breeding.
And now we're gonna go
Far.
some Q&A session because I've got a lot of questions, and I wanted
Dr. Hickey.
you to answer some of them. So one of the questions was, is there an adverse health effect of this technology? So what happens is that when you develop a technology, you do not know what's coming 10 or 20 years after. So should we be concerned about this new technology?
Dr. Hickey.
Yeah. I mean, my frank opinion is no. It's basically growing plants. I think certainly it's quite unnatural to grow plants fast, and there are likely to be changes going on in the genome. There's likely some epigenomic changes, some mutations occurring, just like they're occurring in any generation of plants that are growing. Even
are there any chances of any random mutation? So if there is any adverse random mutation, so that will be devastating for our next
I think so. There's an equal chance of those mutations occurring, just like any farmer's field. But look, if there's a deleterious mutation, the thing is, those plants, once they get to the field, they still need to be evaluated. And if they're not performing, those plants are selected out. They're thrown in the bin, just like any selection process in a breeding program. So, selection works, and we know that. And, it's important that we have large numbers of populations that we are working with in plant breeding. So I don't think there's much concern there. I think when you compare it to other technologies like double haploid systems, there's massive, even chromosomal rearrangements and all sorts of things going on. It's a very intense process, but that system still works very efficiently for plant breeding. So I think, it's really not to be concerned there. I think a lot of the breeding companies, from my interactions with them, have switched from double haploid systems to speed breeding because of the benefits around not only cost, but extra recombination. They've done the modeling over many years and they worked out that would pay off. So that's the main drivers why they switched.
So there was another question. How effective is the technology for rice? So, rice is our main cereal crop in South Asia and some other part of the world. So how effective it is for rice, and can they apply it to all types of crops?
Yeah, what's rice? I'm kidding. Rice? Well, look, I think I didn't mention in my presentation, and it was mentioned a moment ago, that Barry has got some really nice RGA systems going there in Bangladesh for rice as well. I actually did hear about this several months ago. I didn't mention it in my talk. I'm sorry, I should have added a slide on this. Look, I think, and the protocols that have been published and based on Erie's experience as well, it seems like you can grow up to 4 generations in a glass house using RGA techniques with no photoperiod control in some environments in some countries, like Philippines. If you add photoperiod control, you can get 5 generations a year. I know that because that's what Corteva do. It's a similar system to what I showed in millet. It works for rice as well, but you need reliable photoperiod control to achieve that goal. And that requires investment. So you need to weigh up the cost, associated with these things. It costs money to install blackout curtains. Is it worth it to get an extra generation a year? And does it fit into your breeding program? Because it's not just about doing 10 generations a year. You need to fit into the actual field testing season as well, and get all of this lined up in terms of your operations. So there's lots of things to think about and weigh up, and it actually might only be worthwhile to do 4 generations a year. You might not want to do any more than this. So it all depends on how your breeding program is structured and how you adopt this tool in your program.
Thank you, Dr. Hickey.
Now, I'd like to request our current Director General of Bangladesh Rice Research Institute, Dr. Mohammad Shahjahan Kabir, to highlight the research in rice that we have done in the last few decades. So, Dr. Shahjahan Kabir.
And before that, Doctor, we can hear from Dr. Iftekhar. He's the breeding head of Bangladesh Rice.
Okay, so if Dr. Shahjahan Kabir is not present here, so
we will go to
Dr. Iftekhar Jilla. So, Dr. Iftekhar Jilla, if you could highlight some of our research, so what we have done to rice.
Dr. Iftekhar, please unmute.
Can you hear me now?
Yeah, you are audible.
Actually, my Director General is present here. So I don't know why he cannot speak.
Maybe unmute.
Okay.
Unmute.
Okay, it's not a problem. It's
Yeah.
So okay, I can go
first you
first.
go.
I can go first. So this is Dr. Khandogar Iftekharuddollah. Currently, I am holding the position of the head of the Plant Breeding Division, Bangladesh Rice Research Institute. Actually, you know Dr. Gary Atlin, who has already introduced me with Lee Hickey, and I had a couple of email communications with him. And I had a request to him to develop a protocol for speed breeding for rice, because unlike wheat, this is a short day plant. At BD, we are actually scaling up 1.5 million progenies every year following field RGA. But in this system, we cannot scale up strongly photosensitive population, because whether it is single set descent method or pedigree method of breeding, only one generation can be scaled up in the field or in the greenhouse, which has been built following the design of ED, where there is no facilities of controlling photoperiod. So scaling up photoperiod sensitive population is a huge challenge, because this is not only a question of controlling photoperiod, but also controlling temperature and humidity. Other parameters are also important. So actually, what is being done at ED, actually at Philippines, there's no much variation of day length between day and night time, because Philippines is situated in the equatorial region, you know, but at Bangladesh and in this subcontinent, there is variation of day length and day and night period. So photoperiodism or photosensitivity is an important part of product profiles in case of developing varieties, because this is not only the question of strong photosensitivity, but we need to develop weakly photosensitive and moderately photosensitive breeding lines. For instance, in case of field RGA, I think Dr. Lee Hickey is very much familiar with field RGA. He has already shown us a slide obtained from Josh Kopp. It is possible to scale up only 2 generations if we want to develop weakly to moderately photosensitive breeding lines. But strong photosensitivity also an important parameter in case of developing varieties, because after recession of flood water, we need to develop strong photosensitive variety. My director general is always asking me, why not you develop a variety which is higher yielding than BR22, better quality, grain quality with strong photosensitivity. But we don't have any good facility of high throughput speed breeding facility here at BD. So we need actually technical help from Dr. Lee Hickey for setting up a speed breeding facility for rice. So now coming back to the research achievement of Bangladesh Rice Institute, so far we have developed 102 varieties, of which 7 are hybrid and 95 are inbred. And varieties have been developed for different ecosystems, including rain-fed lowland rice, irrigated ecosystem, partially irrigated ecosystem, deep water ecosystem, and so on. Because though Bangladesh is a very small country, but there are a lot of ecosystem variation, and we have a lot of environmental problems, including submergence, salinity, high temperature, low temperature, and whatever you say, we have that problem. So we are feeding around 170 million population. Our population is increasing at the rate of 1.2%, and our arable land is decreasing at the rate of, I think,
1%.
0. Yeah. If we round up it, it is 1%. So, and climate changing is taking place. There's no doubt that climate change is taking place at Bangladesh. So it's a huge challenge for the breeders here to increase the productivity of the different crops, including rice, because rice is the staple food of Bangladesh, and it is taking about 80% of the arable land of the country. So coming back to the presentation, actually it was very much interesting. So my question to Dr. Lee Hickey is that about genomic selection. So do you think that genomic selection is suitable only for prediction of the phenotypic performance in breeds? Can we try to deploy it for the development, for the improvement of polygenic traits like cold tolerance reproductive stress called tolerance in rice. So Dr. Lee, please.
Yeah, I mean, I think that's the trick, isn't it? You can't just apply genomic selection for one trait. You've got to encompass the many traits that you're trying to breed for in a program. You can adopt weighted selection approaches to do that in your prediction algorithms. Look, what I would say is that rice is a similar breeding sort of operations to wheat and barley, and from my experience, the wheat and barley breeding programs, are really using genomic prediction at the stage where they have, inbreds to test. They've developed inbreds, they've developed more than they need, and they apply genomic prediction to that big set to reduce the set down to a smaller number of more elite individuals based on the predictions that enter the field testing program. And so they achieve a lot greater efficiencies because you've already made gain before you've done your field testing. And using the traditional genomic prediction approaches, genomic selection is not the best at identifying the very best parents. It's good for throwing out the rubbish, but it's very difficult to predict the best parents. And so this is some of the methodology development that we're working on at the moment to try to, use that approach to predict the best parents, which I think if we could do that, it would really change how we use the technology in the breeding program. You mentioned that, it's really important, obviously, for your program to operate at a big scale, and you mentioned more than 1 million plants, which is really big numbers. And it seems like you guys are really innovative there, doing lots of things to try to speed up the process, which is fantastic. I guess it becomes a moment in time where, if you've got very good markers for linked to key traits, and then it becomes worthwhile and cost effective to do that marker assisted selection, then you can start to change your breeding operations. And like a lot of these commercial operations, while they do the prediction on the inbreds, they do that very efficient marker screening in F2, F3 for key target traits. And I know, like these breeding operations are still handling numbers in the order or the magnitude of 250,000 plants at F2. It can be done at ultra high density and transplanted into your speed breeding systems, but you need it. You need all these marker screening platforms in place to do it cost effectively. And most, let's face it, most breeding programs just don't have access to that sort of platform. So look, I'd say you guys are doing probably a pretty good job from what I hear in terms of, big numbers for plant breeding and doing it as cost effectively as possible. I think there is room for improvement if we wanted to go down the path of trying to develop the technology more for rice. It's worth exploring because the payoff could be worthwhile, without a doubt.
Thank you, Dr. Hickey.
So with that, now I'm going to request current Director General of BRRI, that's Bangladesh Rice Research Institute, and a Senior Scientist, Dr. Mohammad Shahjahan Kabir, to share his insights into this next generation technology. Dr. Shahjahan Kabir.
Okay, thank you, Mr. Sarkar.
শাহজাহানকে দেখতে পাচ্ছি না তো, just audio video.
Just
Okay, but not video.
Okay, not video.
Okay, that's
okay. Okay. First of all, I would like to thank Mr. Speaker, Dr. Lee Hickey, for his nice presentation. Chair of this panel discussion, Dr. Abdullah Mohammad Sohail, Professor of Biotechnology Division and Genetic Engineering, Jahangirnagar University. Honorable panelist and valuable participant from research and educational institutions, invited guests, representative from print and electronic media. A very good morning. It's my great pleasure to be connected in this important seminar and learned about speed breeding. Though I am not plant breeder, not biotechnologist, I am just statistician and geostatistician and like this. First of all, I would like to share what BRI is doing and how can BRI get benefit from speed breeding, because Hickey mentioned speed breeding can use in any species, even like rice. And Dr. Iftikharullah already discussed elaborately. I would like to share what we are doing. BRI has so far developed 102 varieties. All these varieties have been developed with different special attributes, which contributed to fulfill national demand. Recently, BRI developed rice bison for 2050 and beyond, with an estimated population of 215 million of the country. Another scenario, what I already mentioned, the rice and land of the country decreasing at the rate of 0.4% per year. In the context of changed climate, increased natural calamities, soil degradation, and decreased natural resources. Therefore, alternate options like speed breeding technology can potentially contribute for increasing per unit rice production in order to sustain food security in the country. So far, BRI has breeding program is going with the overall genetic gain, being what we have already calculated, and less than 0.5% in terms of grain yield. There is no doubt that the current rate of genetic gain of BRI's breeding program needs to be enhanced. Since the establishment in 1970, BRI has been following conventional pedigree breeding method to isolate genetically fixed breeding lines. A number of mega varieties have been developed as the output of pedigree breeding method, not only in Bangladesh, but also in India and other rice growing countries. Now the challenge is to increase the rice yield potential for both favorable and stressful environment. Rice varieties should be developed based on the demand of consumer, miller, trader, and of course farmers. Designing of the appropriate product profiles based on the market demand is another important issue to be addressed for different ecosystem. As my understanding, a revolutionary change in the breeding method like speed breeding technology needs to be introduced in order to break the yield ceiling. To implement speed breeding technology, we have started transforming rice breeding through capacity enhancement of BRI. Since December 2015 at Bangladesh Rice Research Institute with the financial support from Bill and Melinda Gates Foundation and technical support mainly from IRI and other organizations and platforms like ICTSAT and the universities and other different outsourced companies like Intertech, DART, Agriplex, Center, etc. The main objective of this project is to enhance the genetic gain of BRI's breeding program through modernization and capacity building. The cutting-edge breeding approaches used under these initiatives, what are developed product profile based on market demand on the basis of product profiles and theory of population improvement, target large size population development to identify expected recombinants involving right plant. Use of rapid generation advance, what we are called RGA, or modified single seed descent technique to develop genetically fixed lines within shortest possible time period. Testing fixed lines in the multi-location trials, even in the earlier yield evaluation, and trials to increase selection intensity and minimize the trial error. Digitalization and automation systems are being used for data collection to increase accuracy. Use of diagnostic high throughput molecular markers for grain quality traits to increase consumer preferences and market price and biotic and abiotic stresses to improve the performance in stress-prone environments. Estimation of breeding values to identify right parents for next cycle crossing program to achieve higher genetic gain. So far, we developed more than 60,000 genetically fixed breeding lines in line stage testing trial. BRI is also scaling up about 1 million breeding population in RGS system, and hopefully within next 4 years period, BRI will be able to develop several millions genetically fixed lines and will be capable to develop superiorized varieties with expected yield 8 to 10 ton per hectare. Shorter growth duration, that is 100 to 120 days in almond season, and 135 to 140 days for boro season. Better grain quality, resistance and tolerant to biotic, that is blast, BLB, false smart, BPS, etc. and abiotic stresses like salt, drought, submergence, water logging, cold, high temperature, etc. By this time, 1,099 LST derived breeding lines are being evaluated in multi-location yield trials. The throughputs for yield traits have been increased by threefolds in 2018 compared to base year 2015. And importantly, multi-location yield trials have been initiated at OYT for increasing selection intensities. I would like to emphasize on the following points for the further modernization of breeding programs. Research management, including centralized rice breeding that will be accountable for delivery of higher genetic gains. Formalization of product profiles that will ensure delivery of varieties with better quality and disease resistance as well as higher yield for specific environment. Parental selection based on genomic estimated breeding values. Engineering support for mechanization, automation and digitalization and their extension to regional stations. Speed breeding for shortening breeding cycle to the further extent. Genomic selection in full operation in breeding programs. Use of bioinformatics for selection breedings. Breeding for reserve, that is B4R, in full operation. Trade development through line augmentation. Development of modern seed drying, processing and storage facilities, etc. So I am particularly thankful to this webinar organization. I expect that the recommendations and decisions coming out from this workshop will pave the appropriate pathways of implementing the valuable concepts of the cutting edge rice breeding technology in BRI and the partner institutes. Thank you everybody to participate and contribute.
Thank you, Dr. Mohammad Shah Jahan Kabir, the Director General of Bangladesh Rice Research Institute, for highlighting some of our achievements in terms of rice research. I have a question that is a common question that we got.
Dr. Lee Hickey.
Are you gonna share your PowerPoint presentation with our participants? So they're asking for the PPT.
Yeah, it's no problem. Or this recording can also be distributed either way.
Sounds better.
And now I'll leave the floor to our event chair, Dr. Abdullah Mawashwal, sir.
Thank you.
The floor is yours.
Thank you, Simon. I am requesting Dr. Tofadzul Islam. He's working on CRISPR. All we know that he's working on wheat. So, sir, if you are here, so please give some of your thoughts on Lee Hickey's presentation, please.
Thank you, Professor Suhail Abdullah and the organizer, distinguished panelists. And I am truly benefited from a comprehensive as well as the state-of-the-art breeding technologies presented by Professor Lee Hickey. really impressive and exciting, and we received different ways of integration of breeding technologies to get to speed up breeding as well as to address new challenges. I am a director of Institute of Biotechnology and Genetic Engineering of Bangabandhu Agriculture University, and we are now working on a serious wheat disease called wheat blast. This disease for the first time emerged in Bangladesh in 2016, and the yield loss for this disease can be 100%. And it is Bangladesh is only the affected country in Asia. It has been in South America since 1985, Brazil, Argentina, Paraguay, and Bolivia. We are trying to develop blast-resistant wheat by using CRISPR-Cas gene editing technology. I am very much excited to learn the express edit technology that described by Lee Hickey, taking CRISPR outside the laboratory and do CRISPR in planta. It is very exciting theoretically for me as a worker, because we are facing serious problem in tissue culture as of our locally important varieties, as well as editing of wheat, because wheat is very complicated due to hexaploidy. My question to Lee Hickey, is it possible to do in planta CRISPR for editing some S genes, that is negative regulators of the wheat blast disease, and is it possible to integrate your speed breeding technique with the gene editing, that is CRISPR gene editing technology, for having faster blast-resistant wheat? We badly need it, because wheat is our second important major food crop, and the day by day, due to blast disease is spreading, our wheat acreage is shrinking, and it is a serious problem, not only for Bangladesh, maybe in future in Asia, because India is the second largest wheat producing country in the world, and China #1 wheat producing country in the world. So if we find a solution of wheat blast, it would impact hugely in the future food security of Bangladesh, as well as in the region, as well as in the world. So I would like to your comment, as well as, is there any scope for you to support us for having for speed up our effort to get the blast resistance in our locally available cultivars. And I finally thank the organizers very much, especially the panelists for a fantastic discussion, especially from BRI. BRI is our #1 most important research institute in Bangladesh. And obviously, my sincere thanks to Lee Hickey for a very wonderful presentation in the in this pandemic, COVID-19 pandemic period. Thank you very much.
Yeah, thanks a lot. And yeah, I think it's definitely an important trait. I've heard a lot about the blast disease there in Bangladesh, and it would be a great one to target with CRISPR. It would be very similar to our plans to target yellow spot disease, where you have sensitivity genes or susceptibility genes to knock out. It's like the perfect targets for CRISPR. Very easily done theoretically, but the technology and methodology that we're developing at the moment is really hugely experimental. We're already developed elite lines that are CRISPR ready, but we're working on testing different delivery systems. So And I believe that many groups around the world are also testing different strategies to deliver guide RNA. One of the techniques that we're using is a nanotechnology developed at UQ, but we'll see what works the best and what's the most efficient. It really has to be efficient editing to really replace the lab as well. And on that note, I might say we've got some really exciting results coming through on accelerating tissue culture by optimizing the tissue culture protocols in a similar way we've been doing the speed breeding, optimizing wavelengths, conditions for better plant growth. It's the same in a glass house or a controlled environment or a facility as a petri dish. And so this is actually really exciting, doubling the rate of regeneration for most of the species that we've been working on, which is even crops like banana. It's really exciting. So,
thank you. Thank you, Lee. We will have some thoughts from Dr. Parthasarathi Bishwas. Before that, I have a few questions, actually, some queries as well. So, Dr. Lee, plant factory and speed breeding facilities, what are the differences between them?
Oh, sorry, just repeat that again.
Plant factory and this speed breeding facilities, what are the difference actually?
Well,
in South Korea, they are using plant factory right now, South Korea, Japan, because they have some environmental constraints. So they are growing short day, long day, some plants, especially herbs or small fruits or something like that in the plant factory. Similar to your this speed breeding facility factory, optimizing different types of lights, temperature and humidity. So I mean, my question to you is that facility same, just application different, or is technology also different?
Do you mean like vertical farming type of systems?
Not vertical farming. This is called plant factory, actually.
Okay. Oh, I didn't know they were calling this plant factory. I thought I made up the term, but it might mean something else that other people are doing. I'm not too sure.
Yeah. Vertical farming also is kind of plant factory, I think, because they are hydroponically, they are growing lots of vegetables, especially vegetables, small fruits like tomato.
Interesting.
Okay, then we can go for next question. RGA, actually this stress accelerated process. RGA, this for the rice breeding, ERI, and I think BD also applying this RGA technique. So speed breeding and RGA, what are the basic differences between RGA and speed breeding?
Well, look, in my opinion, they're really the same thing. It's just a terminology thing, like genomic selection, genomic prediction, RGA, speed breeding. For me, speed breeding is just a more catchier name for the technology. RGA is an acronym. Some people like acronyms, so
okay.
I think they're really interchangeable. But like one of the things that we've tried to push with the whole speed breeding terminology is methodology that does not require embryo rescue. I think that's really important for a plant breeding scale of operations. You really can't be doing embryo rescue on big numbers cost effectively. So that's really been a critical point where we've been trying to differentiate these RGA systems. Yeah.
Okay, another question from the audience. Impact of speed breeding on climate change. I mean, right now, Bangladesh, we are facing lots of problems, like right now flooding. Sometimes lots of these, so many stresses are combating actually. So speed breeding, how actually help to combat those climatic conditions or climatic this so many audience they asked this same question.
Yeah, well, look, in some cases we just need more productive crops, and that just means breeding more efficiently and faster with the elite germplasm that we have to push genetic gain. But in some cases with climate change, it really means new environments for these crops and new stresses that don't often or frequently occur. And to really manage that and reduce the risk for farmers, we really need to, in some cases, be adopting new alleles or new traits. And so they don't exist in elite germplasm. And that means we have to go back to land races or wild relatives to source those genes. And so I think speed breeding can play a really important role there in that pre-breeding and trait integration process to bring those new traits in that we do need for climate resilience. I mean, so it doesn't take 10 years to introduce a new trait from wild material. We can do it in just a year or two.
Okay, another question is very much related to our topic of this session. This how speed breeding will help expand crop diversity, and I mean, how can it help to feed 10 billion?
Well, crop diversity, yeah, again, look, it goes back to getting new traits and alleles from wild materials. We can perhaps speed up the rate of development for crops that have been utilized or under invested in terms of the breeding. A lot of dollars have gone into breeding corn and soybean and then a lot of resources into wheat and rice, but a lot of other crops that are very useful for our humans' diet, they haven't really been invested heavily in terms of modern plant breeding. So perhaps these tools and technologies like speed breeding and other techniques, genomic selection, CRISPR, can help get those crops up to speed and make them more productive and efficient. And as we head into dealing more and more with climate change issues, this will become more important as well.
Okay, and another question that also have come from so many attendees. How much setup cost? I mean, in small scale, can we actually adopt in Bangladesh? I mean, can we bear those costs, or can we make low cost speed breeding system?
Well, I think the answer is yes, but you got to think out of the box and how you set up a facility. You got to think about what are the operating costs. Can you take advantage of existing infrastructure, or are you better off, setting up a fully indoor system inside or in a warehouse or something? It all depends on where you are and what type of climatic conditions experience. You saw some of my examples with shipping containers that can be adapted. That's pretty low cost. If this facility, I think costs about, it was about 20,000 Australian dollars, this shipping container facility. But that included the very best LED lighting systems you can buy from Europe, very expensive, $1,000 each. If you source locally manufactured LEDs that are still suitable for plants, you could really bring that cost down, source local materials and build it yourself. I think you've got to be creative and adapt something that's going to be suitable for your program.
Okay, I am curious about that. In our country, LED, like 3 feet long LED, we can buy only $5. So will it work?
Well, you got to look closely at the wavelengths and the spectrum. Make sure that they're enriched with blue and red. You can have a look at the wavelengths that we've published in terms of the lights that we use, at least across the John Innes Centre and the University of Queensland. If you can try to find a light that mimics these types of patterns, I think that's your best bet. But yeah, always check the light spectrum. The quality is really important, especially if you have a fully enclosed speed breeding system rather than a glasshouse system. In a glasshouse system, you're getting the natural light during the day, so light quality from your LEDs is really a lower priority. It's extending the photo period. You can get away or compromise on light quality in that sort of situation.
Okay, how about if we do in a closed system like tissue culture growth chamber or plant growth chamber, something like that? How about that?
That's when light quality is and light intensity is critical. Yeah.
Okay, so in that case, we have to use this costly LED facilities which have exact wavelength or something like that.
Yes, you know, but it's worth exploring. I know in some countries there are low-cost options, and these look, LED lights are rapidly evolving and becoming cheaper and cheaper by the day. Look, what's possible now was not possible a few years
before,
ago. In terms of light intensity and quality, like it just wasn't there, and now you can grow plants much more cost effectively indoors. The technology is always evolving. The lighting industry is really crazy.
Okay. Next month on 5th, George Lomonosov from John Innes Center will be our guest to speak about plant expression system. So we can hear much more about these facilities, how it is helping to produce plant-based vaccine as well. Okay, now I am requesting Dr. Partho Sharathi Bishwash. He is from also BIRI, and he is working on genetically modified crops. I mean, genetically modified rice. So we would like to hear some thoughts from him. Dr. Partho.
Thank you very much for allowing me to talk here. Actually, in my talk, I want to echo with Dr. Iteghar regarding the RGA facilities, what we are doing under transforming rice breeding program. In BIRI, we are not new actually in RGA system. We have developed a few varieties already, but for developing those varieties, we use this technique for breaking actually the photoperiod sensitivity or something like that, or sometimes hastening the flowering time, covering the plants with black cloth, something like that. But when TRBS starts, we used this technique in our RGA greenhouse, and under this situation, we can now do something like that 2.5 generation per year. But you already heard from Iteghar that we are doing field RGA also. In field RGA, actually we can do usually we did one season per year when we followed the pedigree system, but nowadays we are following 2 seasons a year. But if we do closely, then 2.5 seasons easily we can do. But the technique what Lee Hickey presented here, if we use that one, I guess this we can speed up this process and up to 5 cycles a year. That will actually that will make us a great achievement for shortening the breeding cycle, side by side recycling of the parents, aiming to enhance the genetic gain by accumulating favorable alleles that leads for the yield. And I want to mention here, we are actually tracking genes through using forward breeding system, like different trait markers we are using, especially when the lines are fixed, as we call the line stress testing after the line fixation. In that stage, we track the genes. We actually employ the marker assisted selections for the specific trait markers, and also we are working for at this moment for applying genomic selection. Especially we use AgriPlex platform to fingerprint our genetically fixed materials, around 1,000 markers we use, and we will predict our materials for their performance and select the best parent for recycle them. These are the things what we are doing now, but I think this will be faster when if we have any facilities that like speed breeding protocol that will actually accelerate our RGA system at this moment what we are practicing. I think that will shift. That will be a frame shift for the progress targeting to achieve genetic gain. Thank you.
Thank you, Dr. Partho. Now I have two realistic questions to Dr. Lee. So as Professor Tafadzul Islam and also Dr. Iftakharuddollah and so many curious mind also asking the same question. I mean, how we can get benefit from you, Dr. Lee?
Well, I just gave this seminar. Well, yeah, of course, I'm happy to help where I can. And look, if people there are interested in trying to establish facilities, I'm happy to help advise and share as much information as possible. So you know, yeah, maybe
Okay.
that's a good way.
And then lots of students are listening to you. They are very much thankful and curious. Lots of questions already answered. So they have question. I mean, speed breeding, how help speed breeding to build up their future career, either research or even if they work on speed breeding, can they get good jobs or something like that? Can they apply? So will you speak few words towards our students?
Yeah, well, I think, you know, speed breeding is just one of the tools for plant breeding. You know, I think if you want to be a plant breeder, you got to have skills in and develop really good knowledge of all the different tools and technologies, because at the end of the day, a breeder's got to integrate all of those into a program that delivers varieties for farmers. So you've got to be strategic. You've got to know which tools are best to use in different situations to solve a problem and deliver a better product. So yeah, I mean, I think for me, I've always thought of myself as more of a breeder than a researcher per se, because I don't know, that's just the background I've had, and I've been mentored by lots of plant breeders, and I really love it. I love like integrating the different disciplines and talking to different people and solving problems and using the different skills and science, you know, across physiology, pathology, genetics. It's actually, I find it's really cool place to be, because you get to interact with all these different people and really smart people, and they can help guide you to actually do the breeding and deliver a better product. So yeah, I think plant breeding is a good career option. But I'm biased.
Thank you so much. Lots of questions, and most of the questions already answered. Audience, still if you have more questions, you can leave your question in Science Portal BD, and we will get touch with Lee Hickey to get those answers from him. So now Lee Hickey is midnight for him, so we will not disturb him more. So first of all, I'd like to thank you, Lee, accepting my invitation to share your thoughts. I think lots of people's researchers, scientists are getting benefited from your speech, and we will keep in touch with you. I am forwarding to Simon.
Thanks so much for inviting me. It was a pleasure. Yeah.
Okay, thank you, Dr. Abdullah Mohammed, sir. And I would like to thank Dr. Lee. It's midnight. It's around 1:15 a.m. in the morning. So Dr. Lee, I invite you to Bangladesh. Please come to Bangladesh. You may do a workshop in Bangladesh, and we will be benefited from your knowledge. You know a lot about plant breeding and stuff. So we are inviting you to Bangladesh, and with that,
I've been to Dhaka once, but I'd love to come again. So thanks very much.
So you have been to Dhaka?
Yes, once, only very brief trip. So I'd love to come again. Yeah.
Okay, so it would be a pleasure to host you in Bangladesh. So Abdullah Mohammed Shawal, sir. If we do not have any more questions, then we should
End today's live now.
Yes, I think please end because it's almost morning for Dr. Lee.
Okay, so done for today. Goodbye, Dr. Lee.
Okay.
See you around.
Thank you, everyone. Bye-bye.